Thank you for joining today and welcome to the latest event of the COP29 Virtual Ocean Pavilion. I work with the Global Ocean Forum as a research assistant and will be here for technical assistance during today's webinar. There is a Q&A panel available at the bottom of your Zoom screen where you'll be able to submit questions for our panelists. If you need any technical assistance, you may also put those questions in the Q&A as well. We also will have translation services available for the webinar today through Wordly. I will be starting this translation now, and you should see a pop-up message on your screen that will enable you to join the translation, and it will live stream the translation of what our panelists are saying into upwards of 30 languages. So we hope you'll be able to find one that works for you. And let's see, I will be sending this in just a minute here with all participants. Okay, so Wordly is now available to use. If you would not like to use translation, you are able to minimize the screen as well. And with that, I will turn it over to our organizers from the University of Delaware to introduce the panel. Thank you all so much for joining today on behalf of the Global Ocean Forum and Plymouth Marine Laboratory, the co-organizers of this year's COP29 Virtual Ocean Pavilion.
Thank you so much, Katie. Can everyone hear me? I am excited today to present our panel on oceans of innovation, leveraging technology for ocean biodiversity and community empowerment. My name is Dr. Kimberly Aremus. I'm an assistant professor of marine policy from the School of Marine Science and Policy at the University of Delaware. And I'm excited to present our speakers from the University of Delaware today. So we have three speakers. The first is Dr. Aaron Carlisle, who uses isotope analysis and biologging to understand the behavior, distribution, and ecology of highly migratory marine species. He'll be followed by Dr. Matthew Oliver, who uses acoustic telemetry and satellite imaging to predict where ocean organisms live and factors that influence their distributions. And finally, we'll have Dr. Serena Hakarat, whose research combines marine ecology and molecular biology to identify resilient coral for restoration. Today, each panelist will present their research for about 20 minutes, and then we'll end with a moderated discussion. So I invite all attendees to ask their questions in the Q&A, and we will discuss those questions at the end of the presentations at about the top of the hour. So I hope you will all type in your questions then, and we can also respond in the Q&A window directly. All right, and so with that, let's start with Dr. Aaron Carlisle.
Good morning, everybody. Well, morning for me. Thank you so much for having me today and listening to my talk. I'm going to be discussing how we are using animals to better understand marine ocean dynamics and how we can use this technology to better manage and conserve marine ecosystems and species. So let me share my screen here real quick. Okay. Is that sharing okay? Okay. Great. So anyway, getting back to it. So basically today I'm going to be talking about how researchers are using animal-borne tags to understand what's going on in the world's oceans and using this information to better manage and conserve marine resources and ecosystems. So the first thing you'll notice, as always, when you look at the planet Earth is that it's big and blue. So it's an enormous habitat. It's a big open water planet, right? And so it's full of life, it's deep, it's hard to see what's going on. But so if you kind of were flying through space and you would pass by the planet, this is what you would see. It would basically be a big blue marble. And so I like to start my talks, especially in this kind of area, to focus on how big this habitat is. I'm sure everyone here is very well aware of this, but when you think about the planet, if you actually thought about what you would see visually, you'd be seeing this little kind of ellipse in the middle of the map here, and that's going to be the surface area of the planet, right? And so it's 75% water and then 25% land, basically. But if you look at the livable space on the planet, the place where animals live, it's pretty much all underwater. And so that bigger ellipse is showing you the amount of space that's available for life on planet Earth. And so pretty much 95 plus percent of livable volume in the world is underwater. And most of this is in the open ocean and the deep sea. And so these are huge environments. And they are populated by a lot of very important, interesting species, but they're very difficult to understand and study because of their size. And so some of the things like Dr. Matt Oliver will talk about using remote sensing are a very powerful tool. There's certain limitations though. They really kind of just look at the surface. It's kind of a surface deep kind of analysis. The satellites circle the planet, scanning it, and you get an idea what's going on there. But it doesn't tell you what's going on in this huge volume that is underneath the surface. And this area is filled with animals and life, and particularly animals we care a lot about. So, charismatic megafauna, the whales, the sharks, the dolphins, the squid, whatever. And these are just very important, large, upper trophic level consumers that we're interested oftentimes as humans in terms of fisheries and things like that. But they're critically important for humans and also marine ecosystems. They drive the food webs, they keep things moving in the open ocean. But historically, as I just mentioned, it's really hard to know what's going on. This is a problem because the ocean really regulates all aspects of life on Earth. So it provides us with oxygen, it regulates all parts of the climate. Climate change is going to be fundamentally influenced by the open ocean and the deep ocean in particular. We get food from it, we get resources. It makes our climate habitable in a lot of ways, and there's a huge amount of biodiversity in the ocean, right? So there's a huge number of animals that live out there that are very important for ecological and economic reasons. And also regulates a lot of the world's oceans regulate a huge number of critical fundamental kind of biogeochemical cycles. So really nothing can happen in the world without thinking about what's going on in the oceans. But this is such a huge environment that it's been historically incredibly difficult to study. It's so big, it's hard to get there, it's hard to see what's going on. It's really hard to know what's happening at any given time. And it's also because it's so big, it's hard to enforce any regulations or anything we might have in the open ocean. So, kind of just the nutshell, kind of taking this all away, is that these healthy marine ecosystems are really critical and fundamental to global health, global prosperity. And we, as researchers, as marine scientists, are trying to work harder and harder to better understand how the marine ecosystems work in order to maintain the and sustainable use of marine resources. And so one place that I'm very interested in is using animal-borne tags, so small little computers that you put on animals. You let the animals go and they swim away, and you've tracked them, right? And so it's been over the last decade or two that there's been incredible advances in technology, miniaturization of processors, computer chips, boards, battery power has gotten better that has allowed us to start putting tags on pretty much everything that lives in the ocean. And so you can see in these pictures, everything from bluefin tuna to jellyfish to seabirds, we are putting tags on. And so you can see in that map on the top, this is a subset of available animal telemetry, so track data for a variety of different species from around the world, mostly focused on the Pacific Ocean. And so while we used to look at the ocean, it was a big blue, empty, vast kind of habitat. We are now starting to kind of peel back that curtain and see what's going on in the open ocean, in particular through an animal's perspective. And so you can't really understand and protect any kind of ecosystem or species unless you know where it is, right? You don't know where, if you don't know where it lives or where it spends its time, you can't know how to protect it, in particular from anthropogenic stressors such as fisheries or exploitation. This is some tracking data that were from that study I just showed before with all the points all around the Pacific Ocean. And you can see that over time, you start to look at these animal movements and you start to identify kind of consistent migratory hotspots, migratory corridors, hotspots of activity, places animals aggregate in large numbers. And so these are all new dynamics that we're starting to figure out and learn about just through using these electronic tags. And so these tags tell us all kinds of things, right? And so they measure data, they provide us with data on a variety of different axes or dimensions. So first of all, they tell us the horizontal distribution of animals. So you can see here in the left side, this is some tracks of cod from the North Sea or off around Iceland. You can see in the middle panel, leatherback turtles moving across the Atlantic Ocean and returning back to their beaches in the Caribbean. You can see some tiger shark data, tracks around the Hawaiian Islands over on the right, but you can see where animals distribute them on a two-dimensional basis around the world's planet. You also understand and learn about where they are in the water column. So how much time do they spend at the surface or how much time do they spend at depth? This is obviously very important because depth is the habitat for these animals in the open ocean. So you can see a variety of different vertical tracks of sharks, mantas, and a variety of different animals, showing you how you can learn where they're spending their time in the water column itself. And so obviously, if you're interested in managing or conserving a species, if you don't know if it's spending time at 1,000 meters or at the surface, you're not going to be able to protect it from different stressors or activities, especially anthropogenic human-driven activities like fisheries. In addition, it lets us understand these dynamics through time. And that's also very important. You know, time is a critical axis in understanding how nature works. And so you can see here on the left, you have a basking shark and some squid moving up and down. And on the y-axis on the side, you can see depth down to, 200 or 400 meters, and the kind of darker areas are showing at nighttime. And you can see that for the squid, for example, in the lower left panel, they're up near the surface during the night and they're down deep during the day. You can see things over long periods of time. There's some bluefin tuna over on the middle panel. You can see they're moving up and down the west coast of the United States. That, again, is also fundamental basic information. And so if you don't know where they're spending time, you can't derive or develop important and useful conservation and management strategies. And so, again, if we think back, the open ocean used to be just a kind of a blank slate. We didn't really know what was going on. This is showing some more tagging data, and you can start seeing how much we're learning about what's going on in the open ocean. And this has really revolutionized our ability to understand what's happening out there. And you can see in the bottom line of panels, like different types of animals, tunas, pinnipeds, so seals and elephant seals, things like that, sharks, birds, marine reptiles, whales. You can start seeing how these animals are distributing themselves, how they're partitioning the marine environment. And then you can start to ask questions about the why. So why are they going where they're going? Why are they moving where they do? Why have they evolved in this way? And so with those tag data, the ones I just showed you, you can start to really start diving in and looking at where are animals writ large. So you can see in the upper panels, those kind of heat maps showing you where there's a higher density of marine species. And you can see it over scales, right? In the California current, which is off the west coast of the United States, and the kind of top panel, and then the whole Eastern Pacific on the top left panel, you can start to identify patterns of site fidelity, which are very fundamental and important. So in the bottom two panels, on the left, you can see that these animals we tag, they move enormous distances, tens of thousands of miles over the course of their migratory cycles, and they oftentimes come back to very particular places. We call that site fidelity. So that's repeated use of a particular kind of habitat. And again, that's very important because that's going to be driving. So things like those big tracks in the lower left panel, those animals are crossing many, many different national jurisdictions, different management regimes, the open ocean where there is none. And so this is step one. And so in the right panel, you can see that some of these drivers in the top right, you can see those, that's going to be chlorophyll A, so the amount of plants and productivity in the ocean. And the bottom axis, the x-axis is time of year. So 2 to 12, which is the month 2 to month 12, December, February to December. And you can see these, and you have latitude on the y-axis. And so you can see if you look at the top of those panels, that's going to be up in the northern part of the California current on the, again, west coast of the United States. And the top panels are showing chlorophyll A, the amount of plants that are in the water that are the productivity that drive marine ecosystems. And the bottom is showing temperature. You can see the seasonal cycles where it warms up during the summer and fall, and then it cools off during the winter. And these animals are moving north and south rhythmically with the seasons, following patterns of productivity and temperatures. And so that's going to be influenced by where they find the food they need to meet their energetic requirements to, spawn, migrate, survive, basically. It's also going to be driven by their animal physiology. So every animal has a certain kind of niche, like where it likes to be in terms of the environment, whether it's warm or cold or salty or less salty. And so these dynamics are what we're learning are driving these large scale movements across species and over time. And so once you understand where animals are, you can start to foster effective management strategies and conservation strategies to protect these resources. So here, again, on the west coast of the United States, you can see in the left panel, this is showing you a distribution of potential stressors. So fishing effort, pollution, human activity, traffic, boats, shipping, things like that. And on the right, you can see this information on where animals are spending time based on tags. And you can then kind of combine those to come up with an estimate of where are these animals actually vulnerable? Where is the highest risk to given species? And some of these species are highly are threatened or endangered or of conservation concern. And that allows you to start to develop strategies to protect them. And in this the lower left panel, you can see some of those little polygons, those little shapes off the coast. Those are marine protected areas. You can see that those are being implemented, designed, and put in places where animals are spending a lot of time. And the goal is that that's going to help conserve those species and the ecosystem services they provide. And this is another example. I'm particularly interested in sharks. That's a big part of what my research program focuses on. This is a project that I was involved with, and this is taking a global view of where sharks are spending their time. You can see in the left panels, these are different species of sharks, where they move across the world's oceans, where they're spending time. And so in the upper right panel, you can see that you can kind of condense all these data into where are sharks, period. Like the red colors, the warmer colors are showing, yep, there's a lot of animals there. Cooler colors are where they're not spending time or not as abundant. And so in the middle panel on the right, you can see this is fishing effort. We're getting better at understanding where animals are, fishing fleets are spending time using satellites and other kind of tools. And you can overlay those on top of each other to get what we call a fishing exposure index. So the hotter colors are showing you this is where sharks and fishing fleets overlap, and this is going to be where the risk is highest. And then you can start to manage those spaces for the conservation of the species and the ecosystems that occur in those areas. Another thing I'd like to touch on is that animals we're excited and interested as a biologist. I'm very excited about learning about these animals. But we can also use their behaviors to learn more about how the ocean actually works. And so another way that researchers are using these animal-borne tags is to provide information on oceanography. So what's going on in the world's oceans? So again, oceans play this fundamental role in our climate. In an era of climate change, the oceans are incredibly important. And we need good information of what's happening in the world's oceans over time to be able to understand and model these processes. But again, given the scope and the size of this environment, it's really, 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 really hard to do. So one thing that is becoming a developing area of research and data collection is using these animals to collect oceanography. And so you can see in the bottom right, that's showing you a distribution of tagged animals over time. And these animals, because they're so mobile, they're great at moving around the world's oceans and providing us with information and data about what's happening in the ocean they're moving through. So there's a great opportunity to use the natural behaviors and migrations of animals to better study the ocean and understand and model things such as like hurricanes or monsoons or a tsunami, whatever it is, any kind of oceanographic process. These animals are out there moving through these processes and these dynamics, so we can learn in real time about what they're doing. Here's some example from southern elephant seals. So you can see in the upper right there, these big blubber balls of adorable fuzziness that we put tags on their head and they swim around. And what you're seeing here is these animals are tagged in Kerguelen in the Southern Ocean, and you can see the tracks as they move away from their kind of foraging or their colony where they spend most of their time. They move out for months at a time in the open ocean, diving from the surface to 1,000 meters. And these tags have these ocean observing sensors on them. And so you can see the color, it's color coded by water temperature. And so temperature is fundamental to climate. And so understanding the water column properties of how warm it is or how cold it is really important. And so these seals are swimming around doing what they're doing, and they're providing us with basic information on how the world's oceans are doing. And the idea is pretty simple. You tag them, and see over on the left side, they're out there swimming, they're moving up and down the water column, continuously doing what they are, which is the perfect way to observe what's happening underneath the world's oceans. And every time they come up to the surface to breathe, the little tag on their head is talking to satellites and downloading data. And so you end up collecting real-time information on where these animals were and what's going in the water column, where those animals are over time and space. And this then gets fed into a variety of different end users and stakeholders who are interested in these data, get used in regional oceanographic models or global oceanographic models, used for predictive purposes, all kinds of things. So it's incredibly important and useful information. Another thing that I think is very exciting is we're actually starting to use animals to survey what's happening biologically and ecologically in the world's oceans. So with the advent of a lot of new technologies, this is showing you, so on the right side, this is a gray reef shark that we tagged in the Indian Ocean with a camera tag. And you can see the shark there on the left, and the tag stays on and basically records what this animal is seeing. It's giving us the animal's perspective of what's happening in the world's oceans. And if we want to understand these dynamics normally, we drop someone in the water or take pictures, whatever, and that disturbs animals, disturbs the natural order of things. So you can just start dropping these cameras on animals that are part of the natural environment, and they can go out and see what's happening in the environment. And that's, again, very fundamental basic data, way cheaper, more efficient way to collect these data. And you can start using this to categorize and quantify what's out there. So you can see here on the left, this is showing you two different types of habitats the shark was looking at, the reef flats kind of up on the top of the reefs and the reef walls on the bottom. And then this just diversity of fishes that we identified on the right. And this is all without humans being in the water. You see way more things when there aren't humans in the water because humans make a lot of noise, blow bubbles, disturb things, and animals change their behavior. So we're getting a very different perspective. This is a similar camera tag on a white shark off South Africa, swimming through a kelp forest. That's the head of the shark up in front. And you can see that we're using this to recreate its track. You can see in the middle panel on the left, as it's swimming through the kelp forest, we can use it to count the density and types of kelp and habitat, where they're seeing other kinds of animals, including other white sharks. You can see in the bottom left panel, a white shark in the distance, these interactions. And you start to see very different perspectives of what's happening in the world's oceans. In addition, and finally, we can use these newer tags to understand and quantify not just what the animals are seeing, but also what's going on in the ecosystem. This is some amazing data that's been kind of coming out recently with elephant seals again, where they're putting these camera tags on them. You can see in the lower left panels, you can see these foraging events where these animals, this is in hundreds of meters of water in the open ocean, where they're coming across different types of deep water animals and they're eating them. And because they're coming to the surface, you can overlay, and you can see in the top right panel, by color, like where they're actually foraging and where they're encountering prey, which allows us to then model and understand where in the open ocean are things going on hundreds or thousands of meters underwater. Where is the life aggregating? Where is it? And this is, again, very new, fun basic data that we've just never seen before. And this is only in the last few years that we're starting to see some of these dynamics. And so with that, I think my time is over. I just want to leave with the kind of basic statement that these new constant advances in technology are always going on. As an exciting time for ocean science and marine research because we are starting to see things for the first time. This is a wonderful video taken right off here in the mid-Atlantic, showing some spinner sharks swimming around together. There's a camera tag on the dorsal fin of the sharks. And again, we would never see this without these tags. So with that, I will sign off and I'm happy to take questions later on.
Thank you, Aaron. And next we have Dr. Matthew Oliver. Dr. Oliver, do you need, perfect.
Okay, you guys seeing the correct screen now?
Yes.
Okay, all right. Thank you, Aaron, for that. And it was actually a really nice segue for what we've got going on here. Oops, hang on a minute. I've got some Zoom things in my way here. There we go. Okay, so yeah, my name is Matt Oliver, and I am a professor of oceanography here at University of Delaware. And Thanks for that, Aaron. That's actually a nice segue to what we're doing, talking about the remote sensing of biodiversity for conservation. So um, I just want to give a big shout out to my collaborators here, Dwayne Fox from Delaware State University, Matthew Brees from St. Mary's College, Daniel Halsey at Hubbs Research Institute, and Robert Roos at the Still-Guamish Indian Tribe. So this work that I'm going to show you has been funded by multiple agencies, mostly by NASA, because our focus has been on taking some of the data Dr. Carler was just talking about and fusing that with remote sensing data and developing models. And so our motivation for this really has been I think this is a great paper for you guys to take a look at from David Tillman. And he says that the world that will exist in 100 and 1,000 years will unavoidably be of human design, whether deliberate or haphazard. And so our choices are either to organize and create the world that we think we're going to need or want, or we're going to have a variety of anthropogenic forces doing it in an uncoordinated way. And of course, one of the consequences of that, this is the Living Planet Index on the right, which measures the average decline in monitored wildlife populations. This is terrestrial and marine, and we've had just a dramatic reduction in the last 50 years, and it has no sign of going back so far. And so we do need coordinated efforts in order to reverse this, specifically in the marine environment. The principles that guide this design that we're going to be working on must be based on science, of course, and much of it is only done sketchily to date, and on ethics. And this has been actually a big push in the NASA world. I don't know if you know that NASA funds quite a bit of biological diversity and ecological forecasting, and this was a great report that came out really focusing on the emerging technologies and remote sensing and what we can do in the biodiversity space. And so this has caught the attention of many places, and of course NASA is playing a big part in this, and the research I'll be showing you today is funded primarily through them. Of course, there are consequences when you put forecasts out there of where you think biodiversity or species are. And so there are ethical considerations. And when we put forecasts out there that are based on remote sensing satellites, we need to make sure that they apportion costs and benefits between individuals and society as a whole. and between current generations and future generations. And so some of the work that I'll be talking about was highlighted in this paper about the unanticipated consequences associated with ecological forecasting driven primarily by remote sensing. And of course, a sustainable world, and this is still from, these are Tillman's quotes here. A sustainable world will still require an ethic that's ultimately as incorporated into culture and as long-lasting and as constitutional Bill of Rights or as religious commandments. And the point that he's making here is that these abstract concepts like sustainability or biodiversity conservation are only as strong in the commitments of people, not really nations, but the individuals who are actually out there acting in the environment. Most, quote, sustainable decisions in the ocean are done away from the gaze of the law. We don't have a lot of enforcement in the open ocean at all. Even in the coastal ocean, like in federal or state waters, there's some, but a lot of it is really up to the individual acting when nobody is looking. And so one of the things that we think about in our research is actually what motivates conservation practices writ large, and how can we change either that motivation or work with people that are maybe have low motivation for conservation practices. And so what I'm going to show you in the next couple slides is just an overall framework about how people think about the motivation for conservation. And then we'll get into some of the details about how we're addressing a particular group using some remote sensing tools. So if we just have an overall relationship between environmental concern and wealth, I would say there's two major theories, although they compete with each other. One is called the materialist theory. And this idea is that as you get wealthier, your general environmental concern gets lower. And the reason for this is that poor communities lack the infrastructure, wealth that generally insulate themselves from the environment, like water quality. And so as the wealthier communities become, the more insulated they are from the immediate impact of the environment, and therefore it's less concerning. And so they're not tied directly to the day-to-day quality of their environment. Countries that are in this situation tend to have more extractive economies. They're heavy miners, heavy fishers. And the main driver of concern for people are sort of day-to-day resource issues. And so they're really struggling on a day-to-day issue about what sort of life that they want to have, where their kids are going to go to school, do they have enough food, things of that nature. And so this is the general expectation. That is over against another theory, which is called the post-materialist theory, in where people believe that as you get wealthier, your environmental concern increases. And so the idea is that the richer you are, and since your basic needs are met for food and safety, this group develops a set of luxury concerns that operate on space and time scales much larger and longer than day-to-day concerns. And so once your basic needs are met, you can start to worry about things that are going to happen in faraway places from you, or decades or even centuries away. And so you have the freedom to worry about these larger problems. These tend to be more service-based economies. And of course, the main driver for concern is successive generation. What's your future going to be like, not just on Tuesday, but in 100 years, maybe for your grandchildren. And so these are two basic ideas. And one of the big questions that comes up in the discussions between these papers, and I have some papers to read on my last slide that discuss this, is what do the middle income people believe? And middle income is largely lower and middle working class users, and they generally are expected to have the least amount of environmental concern based on the intersection of these two ideas before, not always. So if you go to the Gapminder, and you can pull up some interesting plots here One is income versus this is a Gallup poll that see climate change as a threat. Maybe you can see some of the hints of these. I've highlighted some of the bigger economies out there: Japan, Germany, USA, India, China. Maybe there's a weak relationship as income goes up. This is per person GDP per capita, inflation adjusted, that as maybe they get richer, the they see the threat, or they see at least see climate change as a threat. So that's maybe an index for their concern for the environment. And maybe there's something over here where you've got the weakest countries, or at least the poorest countries, I should say, maybe on a materialist curve. And so maybe that's there, maybe that's not. But you can ask that of whether or not that commitment leads to action, right? And so this is now income versus the terrestrial and marine protected areas percent of the total area. And one thing you can see is that maybe on average, the richer you are, the more protections you have. I also have Malawi on here, which is a very poor country, but it's also a tourist destination as well. People come and enjoy the environment there. So maybe that's there. But in any case, that the thing that we're going to be focused on here is much smaller scale than those global averages. We are really thinking about what happens when we try and take a group that we think actually has low concern for the environment, which is this middle income group, and we hand them a conservation tool that's based on remote sensing. And some interesting ethical concerns, in fact, start to pop out. So previously, like Aaron showed you a bit about satellite tags. These are acoustic telemetry tags. These are coastal fish that these are Atlantic sturgeon that transmit pings at about 69 kilohertz. And we actually go in and we tag these fish and insert a tag in them surgically, and then they swim around in the coastal ocean. And a lot of fish do not come to the surface to transmit satellite data. So these are ones that are in the coast. These Atlantic sturgeon are endangered in the coastal ocean. And so we can actually see where they are based on their acoustic pings. Now, once we have a record of their acoustic pings, we merge them again with remote sensing satellites. So what you're seeing on the right-hand side are the little dots that are coming up there in Delaware Bay. This is on the east coast of the United States, are where these fish are being detected at any given time. And we can merge them then, in this case, with sea surface temperature. But there are literally dozens of remote sensing satellites that measuring all different sorts of properties of the ocean color, productivity. The new PACE satellite that NASA just launched is going to get down to species levels and community structures of phytoplankton. And so it's really an exploding area of data. And so now we have this really interesting situation where we can merge these two data sets together. We've tagged both Atlantic sturgeon, which are on the endangered species list, and also the sand tiger shark, which is on the IUCN critically endangered. And what you're seeing on the right here are the movements of these animals between acoustic telemetry positions in Delaware Bay. The blue dots that are moving up the river, those are Atlantic sturgeon that are getting ready to spawn. And then the red dots that are coming in, those are sand tiger sharks. And so what's really cool here is that because we have these kinds of observations, we can reconstruct their movement patterns. And just like Aaron showed previously, we can look at areas of high use, see what sorts of areas maybe need to be protected, how they might be targeted. And it shows just a super cool dynamic in between these two animals in the bay. So one thing that we learned when we started to take data like this, both the Atlantic sturgeon and the sand tiger shark, and start making forecasts, especially for people who maybe either targeting these directly or whose activities in the ocean may inadvertently create bycatch opportunities is that we needed to be open and transparent. We needed to work with really diverse stakeholders to understand their needs and their concerns. And so the group of fishermen that we were really focused on with our tool that we created over here, which is called the Atlantic Sturgeon Risk of Encounter. We would put out models that would predict where these fish were going to be at any given time during the day based on real-time remote sensing. And our models were 95% accurate to where these fish were. And so they were extremely powerful to show where these fish were going to be. The fishermen that we were working with in this case were small boat fishermen. They were going out and not targeting Atlantic sturgeon, but they were targeting striped bass, which is a legal gill net fishery. But of course, the gill nets do, in fact, impact the Atlantic sturgeon. Shipping is also a big concern in Delaware for impacting Atlantic sturgeon. But these fishermen here were our major target here for our risk of encounter. These fishermen are low to middle income fishers. They generally are out on the bay without GPS. They have a depth signal, and they are really sort of making a living day to day based on their catch. But one of the big things that we found out is that conservation groups, which are post-materialists, were really concerned that we were facilitating the illegal exploitation of an endangered species. So by making these forecasts public and where we thought these fish were going to be that were highly accurate, they thought that we were probably exposing this endangered fish to exploitation. So the reason the Atlantic sturgeon are very low in their populations is that they have been historically exploited for caviar. And so there was some concern from conservation groups that we were uncovering where those fish were going to be. We were sort of removing the mystery and making them easy targets. The fishermen, on the other hand, these middle and low income fishers, were really concerned that this tool was going to be used to control their ability to make a living. So they saw it possibly as a way that agencies would clamp down on their activities, and they would then be governed by whatever the tool was going to say, and they saw that as a major crimp on their ability to make a living. And so even though the tool was extremely accurate, at first it was not very popular based on these two responses. Now, that's what we heard, but what actually went out is that we created a tool that not only just went out on the web, it also went out to automatic text messaging to show where these fish were going to be. And the reason we did that is that most of our users, which were fishermen, did not have broadband internet access, which would require this, or at least they didn't want to pay for it, which would require this sort of web tool, but they did want to get their information via cell phone. And so they were low bandwidth users, but they did use it, which was interesting. So we put this out, and there's maybe 20 to 30 commercial fishermen that were regularly using this. But if we actually looked at our time distribution of hits and usage during a general day here, this was during migration season, you could see that we were up to 5,000 hits. And so our tool was being spread widely, probably through channels that we weren't putting it down. They were probably forwarding it through their text to friends or something like that. So we know that there's interest, but they're actually quite secretive in how they're using the product because they don't want to be regulated by it. Now, similar to the Atlantic sturgeon, we also made a highly accurate forecast of where we think sand tiger sharks were going to be. That's an example of this on the left. Again, hotter values are more indicative of where we think they're going to be. But our research group, we're post-materialists, we're concerned that we were facilitating the illegal exploitation of sand tiger sharks. And so there is, unlike the sturgeon, where the sturgeon are a bycatch of a legal fishery, we do know there is a directed recreational fishery, which is barely legal, probably not legal, in Delaware Bay, where middle income fishers are actually targeting sand tiger sharks for cheap charters, and they've done this mostly after the collapse of the weak fish industry in the Delaware Bay. And so the sand tiger is big, it's scary looking. You can see this is I'm not showing you who's doing it, but there are family shark fishing advertisements around the harbors here, and it says get to catch something big. And so these generally attract middle and low income fishers that can't afford to run very far offshore and burn lots of fuel to get a big tug on the line. They generally practice catch and release, although maybe some questionable practices there. They tend to serve poor minority communities that are taking these trips, and it's easy to catch a really big fish without burning a lot of fuel. And the cost is generally around $60 per person. Now, we decided to not make this forecast public because we were concerned about this level of exploitation. And so that was an ethical choice that we made, but we did feel like we were picking winners and losers because the power of these remote sensing techniques is pretty great. We did release it to the National Aquarium because the National Aquarium does go out and target these animals to put them into aquaria. And so we did release this to them, and they also serve poor communities, and they charge $65 per person for a quote shark experience. We did decide to give them the shark forecast. We're still trying to decide whether or not that was the right thing to do, because it's quite effective. So one of the things that I think is really interesting about this is that these real-time forecasts of species and/or biodiversity is an emerging disruptive technology. They are coming. There is, because of the fusion of the tracking and telemetry data along with the satellite observations and the development of computer models to do this, and they're also doing this with AI, is that you can expect extremely accurate models of where these protected animals or any animal are in the environment. And so I think there needs to be probably some level of control or decisions about how we decide who gets these types of forecasts. Do we need some sort of biodiversity forecast ethics board or something like that to put this kind of information out? One of the questions we have is how we're engaging the middle income users of the marine environment, the people that are actually out there day to day working on this stuff, whether or not they're able actually to make good decisions when nobody's looking at them. And we're trying to work with them to see whether or not they're going to be more or less environmentally concerned as their economic needs are met. So with that, I'm going to finish up. This is just real quickly the references if you're interested in some of the theory that we were talking about in the papers. And I'm happy to take any questions after the next talk. Thank you very much.
Thank you, Dr. Oliver. I see there's some hand raised in the attendees. I would like to direct you to the Q&A part of the Zoom screen, because that's a great place to pose a question for our speakers. So if you could do that, that would be fantastic. And we will have a discussion at the end of Dr. Ackrott's talk. Go ahead.
Thank you. Can you see my screen okay?
Yes.
Perfect. So I'm Dr. Serena Hackrott, and I recently joined Dr. Mark Warner's lab at the University of Delaware as a postdoctoral researcher. And I'll be zooming in a little bit to the ecosystem and organism scale for my presentation. I'm excited for this opportunity to share our collaborative project, developing an accessible tool to predict coral resilience, and similar to Dr. Oliver, working closely with stakeholders to aid coral management and restoration. Coral reefs are some of the most biodiverse ecosystems globally and provide critical ecosystem goods and services, such as food security through coastal fisheries and coastline protection by reducing wave energy. And of course, healthy coral reefs support multiple levels of the tourism industry. Overall, coral reef ecosystems are valued at billions of dollars and support millions of people worldwide. However, as we are all becoming increasingly aware, climate-driven coral bleaching is leading to widespread coral mortality, which ultimately results in reef degradation and the loss of those ecosystem services. Unfortunately, these marine heat waves and mass bleaching events are predicted to occur more frequently and become more severe with the current climate trajectories. Coral restoration is a way that we can try to buy some time for these populations while we tackle the climate crisis. Propagating corals through asexual fragmentation can help sustain declining populations and provides material to reseed degraded reefs through coral outplanting. And for some critically endangered corals, these efforts are vital for species preservation. However, the coral gardening approach is resource and time intensive, and a major consideration is which coral colonies to include. It's been well established that maintaining genetic diversity should be a priority. But more recently, managers and scientists have discussed the importance of identifying more hardy or resilient corals. Yet this is not always an easy task and might require large-scale monitoring through bleaching events or experimental stress testing to see which coral colonies have the desired traits, such as heat tolerance. And instead of waiting to observe coral bleaching, it would be more efficient if we could know beforehand which corals are more resilient. And just as we might have a family history or a genetic predisposition for certain traits or diseases, corals are also genetically predisposed to display certain characteristics, such as stress tolerance, reproductive success, and growth rate. And this understanding can be harnessed to help select corals more likely to survive in restoration efforts. However, corals are more complex than they look. In addition to the coral animal that we see, the meta-organism is also made up of microscopic endosymbionts, including microalgae, bacteria, and other microbes. These different partners live together in symbiosis, or the mutual exchange of resources. And corals actually depend on the photosynthesis of their algal symbionts for most of their nutritional or metabolic needs. And when this symbiosis breaks down, it presents as coral bleaching or the loss of algal symbionts. And just to make things a little more complicated, there's also a diversity of algal symbionts that vary in their thermal tolerance and metabolic support of growth or reproduction. So the algal community further contributes to that genetic predisposition of the coral organism. Similar to blood tests that you might have during your annual checkup, molecular biomarkers have also been identified for corals, which can help predict resilience. Advances in these molecular techniques have provided detailed insights into the cellular mechanisms underlying coral responses and resistance to environmental stress. Yet they're limited in their large-scale application for conservation. These techniques require invasive sampling, and the cost of sequencing restricts the affordable sample size. The analysis also requires bioinformatic expertise and overall a long waiting time between the sampling and the critical results needed to help select resilient corals. Our approach aims to limit these challenges by developing biomarkers based on non-invasive measures of photobiology, paired with machine learning and cloud-based analysis pipelines to predict coral resilience. These measures of photobiology specifically detect chlorophyll fluorescence. The algae inside the coral cells contain chloroplasts, where chlorophyll is the main pigment to absorb light for photosynthesis. And while most of the light is used for the process of photosynthesis, some is also lost as heat or fluorescent light. Measuring this fluorescence can tell us about the rate of those other processes and the overall photosynthetic health of the algae. This method can even detect the photo damage that can occur under environmental stress, which is an early warning sign of coral bleaching. Our collaborator, Dr. Kenneth Hoadley, has designed a fast repetition rate fluorometer to measure fluorescence within coral algal symbionts. Using different wavelengths or colors of light, specifically targeting the different pigments within the algae. The light given off as fluorescence under these different light conditions is then detected and provides multiple metrics of photosynthetic health, including the activation and relaxation dynamics as light is captured and used by the photosynthetic machinery. The combination of all of these health metrics are what we call the bio-optical phenotype, which together can be used to differentiate groups of corals that fall into a certain phenotype depicted by the different colors on this clustering diagram. This bio-optical phenotype is driven by the genetics of both the coral animal and the algal symbiont, and most importantly, can be used to predict resilience. For example, here you can see those bio-optical phenotype clusters stretched out across the top of the figure. Colored bars represent the coral species, and you can see that the coral species alone does not fully explain those phenotype clusters. However, when the genetic identity of the algal symbiont is added, the combination of both coral species and the dominant algal symbiont better explain those patterns in photobiology. And many metrics of photobiology are correlated with thermal tolerance, which can be used to build predictive models. Here's an example where such a model was built and validated for Caribbean acropora corals, showing significant correlations between the bleaching severity predicted by the bio-optical model and the degree of bleaching that was actually measured. Our project builds upon this framework, where first corals are measured to capture the variation in both photobiology and thermal tolerance. Next, machine learning is used to develop models that can explain the relationship between the bio-optical phenotype and the bleaching response. And finally, those models are validated by comparing the predicted bleaching outcomes to those actually measured. But so far, this method has only been applied to a handful of coral species. And as I mentioned earlier, coral reefs are incredibly diverse and made up of many functionally different corals. So for this method to be widely applicable, it's important that it's accurate across this diversity. The new project in which I'll be working aims to address this gap, where we will optimize and scale up these bio-optical tools and aid in the ongoing reef restoration and management by our Pacific partners. This project is funded by the Coral Research and Development Accelerator Program, or CORDAP. So to scale up this approach, we're evaluating over 10 new species and collaborating across 3 countries. And we're building models based on both experimental and natural bleaching responses. And we're also comparing the accuracy of models across seasons, regions, and species to identify the most widely applicable model. We're currently in the first year of this 3-year project, where we're collaborating with restoration programs in Fiji and Guam to pair laboratory-based measurements of photobiology and short-term thermal stress testing with field-based experiments and monitoring through natural bleaching events. We're assessing variation in photobiology and thermal tolerance across a wide range of species and under different environmental conditions to increase the accuracy and broad applicability of our bio-optical models. And we're also measuring longer-term growth to see if bio-optical models can be used to predict other critical fitness outcomes. Here I'm sharing some preliminary results from the research expedition that I joined in Fiji this past summer and some of the data that I'll be analyzing during my postdoc. So the points on this figure represent the photobiology of a single coral colony, and points that are closer together are more similar, while points that are farther apart are more different in their biooptical phenotype. We're seeing differences in photobiology across coral functional groups, represented by the different genera shown in points of different shapes and colors. And in this next figure, we can see further clustering of each coral species shown in the different colors. We're currently analyzing the environmental factors driving differences across different sites, and we'll also add the genetic identity of the symbionts to this analysis. Some of the major limitations in the other methods to predict coral resilience were the need for analytical expertise and the long lag time between the sampling and results. We aim to circumvent those obstacles by providing cloud-based analysis support on a Shiny app designed by Dr. Hoadley. This site will allow users to upload biooptical data collected from a range of corals for an automated cluster analysis and predictions. This provides a rapid turnaround of results that predict coral thermal tolerance and resilience, and allow practitioners to identify which colonies to target. The goal of this pipeline is to reduce the need for analytical expertise and quickly provide the results needed for decision-making. And one of the most important outcomes of our project will be the capacity building through technology transfer. Our immediate goal is to train our restoration partners on this method throughout this research and development phase. For broader use, Dr. Hoadley's lab is also working to minimize the cost of the instrument, which is already less than a quarter of the price of commercial instruments. And will also optimize the cloud-based analytical resources as we continue to build and validate models through this project. This project is a large collaborative effort across universities and countries, and I'm grateful for the opportunity to represent this group. The names listed here are those involved in the most recent research expeditions, but this work also builds on years of initial research, and you can see those acknowledged on the pilot version of the Shiny app that's linked here with this QR code. But please note that the site is still under development. And I've also added my contact information at the bottom of the slide in case anyone's interested in potential collaborations or would like any additional information, especially since I will warn you now, I am not the machine learning modeler on the team, but I'm happy to answer any questions or put you in contact with our team members for any that I can't answer fully. Thank you for your attention.
Thank you so much. That was such an interesting panel on technology around oceans and data that's being collected, ethical concerns, concerns about technology transfer and capacity building. So now is a great time to open up this panel to questions from the audience. So invite the panelists to turn their cameras on. And I will start with some of the questions we're starting to get in our Q&A now. One of the questions I had was around where are still the gaps in data? And there's a specific question on this for Dr. Carlisle. Are animal tags being used in any deep sea animals to explore lower sections of the ocean?
Oh, that's a good question. Yeah, I mean, so the deep sea, not so much, just because there's so many challenges to deploying these types of tags at those depths. Oftentimes, things start getting crushed after you get about 2,000 meters or so. So this deep sea, there isn't really commercially accessible tag at this point. And also the big problem is Dr. Oliver mentioned is like getting the data back. So if the tag is in the deep sea, how are you going to get the data back? And so sometimes we have tags that will pop off and float to the surface. Um, and there has been some of that done, but again, it's hard. Just a more challenging environment. The deep sea is just a hard place to work. But that's definitely something that is in the kind of infancy, but it's advancing. There are efforts made in that kind of direction right now where they're trying to build new tools, get better technology to deal with the pressures associated with the deep sea.
Thank you. That raises all sorts of questions on sort of where you see technology going in terms of whether it can actually measure things deeper, but also temporally, are there limitations in how long these tags can survive? And is there any advances being made in that direction?
Yeah, so it's all about battery power for the most part. And so it's this push and pull between how much data you collect and how long the tag can function. And again, it always comes back to getting the tag back or getting the data back from that tag somehow. And so there's lots of different ways to do that. Dr. Oliver talked about acoustic telemetry. There's different kinds of tags that are archival, which will just record forever, but you have to catch that fish or that animal again to get that tag back. So there's lots of different ways to potentially get that, but it's ultimately about battery power. And the more battery you have, the bigger the tag is, and the more impact it has on the animal, and it constricts on what kind of animals you can put those tags on. But there's some really fun, interesting new kind of solar tags that are kind of coming online. But it's again, those animals then have to spend a lot of time right at the surface basking to absorb that sunlight. And as anyone who's ever worked in the ocean knows, if you put something hard in the ocean, stuff is going to grow on it. And so you get biofouling, and that covers a lot of the solar cells and things like that, which again kind of is a problem. And it's something that we're always trying to work on. There's actually some very cool new tags, technologies that are using changes in temperature to generate electrical current. And so basically, if an animal's moving from warm surface waters to the deep sea, every time it's going up and down that thermal gradient, it's charging the battery to some extent. But that is still kind of in its infancy, at least for the animal tagging kind of size, the scale we're talking about.
This leads to another group of questions, and that's around the ethical concerns. I noticed in both your slides and Dr. Oliver's slides, there's a lot of discussion about ethical concerns with the research. So we have a couple questions on this. One is for Dr. Oliver, and it's about forecasting data and whether any of it is shared with enforcement agencies to prevent exploitation of species, or do such enforcement agencies even cover this? Is there a gap in ocean protection that you could foresee with these types of data sets?
Yeah, so we've been open with the agencies. We've been willing to provide anything that they are willing to take. Sometimes the agencies, so there's a couple different things that can pop on depending on which agency you're talking to. One is that if they are provided with the information, then they're maybe obligated to act on it, and they don't have the budget to do it. And so they are not that interested in it, or they will take it under advisement. But one of the questions that comes up in terms of enforcement protocol is, what is the legal standing of the forecast that we're putting out there? How accurate is it? How was it built? And will that forecast sort of hold up in a court of law somewhere? And so that there's some interesting legal considerations that I didn't mention when we're putting out data like this. Right now, we put them out free and we consider them advisory. Some of this stuff is now going to be challenged in court. And so there are lawsuits that are that have been filed in New York, New Jersey, and Delaware from NGOs that they are not using all available information to protect species like this. And so that's just getting started. It's not clear to me exactly how this is going to be treated in a court of law, but until that happens, it's not clear how an enforcement agency would be forced to use a tool like this. That being said, I mean, Dr. Carlisle talked about the open ocean, and everybody knows that the open ocean, there's hardly any enforcement. That doesn't mean there's more enforcement in the coastal ocean. Technically, there is more, but it is still so minimal in comparison to what you have like in land-based systems like forest rangers and national parks. It's just the cost to get people out into that environment is big. It's expensive to do anything in the ocean. And so because there's boats and people and fuel and weather and all that sort of stuff. So that's one of the reasons why, at least on our approach now, we're trying to change or work with people on their behaviors when no one's looking. And at the end of the day, I think that's the thing that probably has more teeth, right? So if you you know that when you're driving a car, everyone slows down when they see the police officer, right? But what you'd really like to do is have people reduce their speeds even when no one is around. That would be the safest result. And so, of course, I'm guilty of that too. And so that I think we try to have as much working relationship with the users of the environment. I'm sympathetic to the lack of, the concern that there is a lack of enforcement. I'm skeptical that we're going to get a lot of enforcement in the future. And so I do think the approach is to be working with the people who are actually out there every day, interacting with or potentially interacting with these animals.
Just keeping on this topic of ethical concerns, we have another question on thinking about sort of these non-human species we're monitoring, and that in social science, human subject protocols have been critiqued for focusing too much on individual subjects and not enough on communities. Is there a parallel with non-human species monitoring in terms of monitoring individual species versus community of species? And what suggestions do you have for guiding decisions about ethical data collection and potentially release with some of these collection techniques? And this really could apply to anyone on the panel, but maybe Dr. Carlisle might have some initial thoughts.
Yeah, no, that's a great question. And there's a whole idea called ecosystem-based management, which fisheries management, especially in wealthier countries, they've been talking about for decades. But it's really hard to do. How do you measure an ecosystem? How do you know how it's doing? And that's just it's a fundamental trait of the ocean. It's hard, as Dr. Oliver was saying, it's hard to just even see what's happening. It's not like you can use your eyes and see through the water and count things. Counting how many fish are in the ocean just is one of the classic challenges in fisheries management, right? And so it's a very simple thing that it's a hard for most people in our kind of terrestrial existence to wrap our brains around. It's just the ocean is opaque and you can't see what's going on in it. So we have to get this information indirectly. It's hard to do for a single species, much less a whole ecosystem. So you have to think, how do you measure it? What do you measure? How do you get those measurements through time and space in a way that can be scaled to effectively, implement some sort of management effort? Yes, that would be way better to do, and that's where management agencies, researchers are actively trying to get to, but it's really, really, really hard to do, and that's why it's taken so long and is still kind of very much in process, I would say. But that's a great question. I think there's a lot of value in thinking about things in the larger scales and the community level or ecosystem level, but it's just a difficult nut to crack.
I think that's also been a critique for coral restoration, is that it's focused on individual species where we really should be restoring the ecosystem function. And as Dr. Carlisle says, that's multiple species of not only corals, but other benthic organisms and fish to really restore that function. And one ethical concern with restoration, even just focusing on an individual species of coral, is that while we're sustaining these populations, we're also imposing an artificial selection for certain traits. And while something like thermal tolerance might be the more immediate concern, kind of going back to those conflicting theories that Dr. Oliver mentioned, we might need to be considering growth rate, the structural strength of the coral to resist storms, its reproductive output and reproductive success. So a lot of those traits actually come in with trade-offs to each other. So as we're making these tools to select for certain traits, we also really need to inform the stakeholders and the end users about the importance of including other traits in that selection process and still maintaining that genetic diversity instead of only focusing on our favorite performer that might not be doing the best in other circumstances or other traits.
That's so interesting. And that raises some other questions in terms of how do you communicate with these stakeholders when introducing some of these technologies. You mentioned your project is in the stage of training different users how to use the technology. And so have you had any experience with that so far in having these difficult conversations to start engaging them on the complexity of the analysis, what it includes and what it doesn't?
That's a great question. Well, luckily, the partners that we've established this project with are long-established restoration groups, and they have that scientific background as well, and have been thinking about those things for a while, and they're managing their local environments, so they are more in tune than some in thinking about conserving their own environments. But with restoration becoming more popular, kind of a fad almost in conservation, there's also been discussions on who is allowed to do coral restoration, and is there any sort of regulation on selecting the sites. There, of course, you have to get permits, and it's usually limiting that way. But just also checking the plans for restoration and how they're selecting the amount of genetic individuals to include or the locations. That's not really as regulated, especially across the world. So I think that that's definitely a major concern as we're expanding to newer restoration groups.
I would like to, at this time, note to the attendees that you can put your questions in the Q&A box at the bottom of the Zoom link. So hopefully all of you have access to that. So please continue posting these wonderful questions. Your thought, Dr. Hackerott, on sort of who can do the restoration is very interesting. It also reminds me of what Dr. Oliver brought up in terms of who has access to the data to even begin with. I was wondering if any of you have, in your experience so far on the ground working with these stakeholders, tried to measure in any way the responses from sort of giving out this information. Has it improved, for example, sturgeon populations since you've started making some of this data available? So I'll direct that to Dr. Oliver.
Yeah, that's a great question, and that's one that we get asked a lot, of whether or not it's actually improving the situation. I would say that there are two reasons that we don't know that. The first is on the agency side, because some of this stuff has not been challenged in court. We started doing this in 2015. Because it hasn't been challenged in court, or at least mandated through federal and state systems, it's been through sort of volunteer action only. That might change now with several pending court cases. It's hard to know the direction that's going to go. So that's kind of like the the stick, approach. The carrot approach is that we do have responses from our fishers that they're actually the ones using the text-based messaging, not so much the web app. And they have given us feedback, of actually quite voluminous feedback on the types of messages that we're putting out. They haven't told us very much about whether or not it changes their behavior. But I would imagine, so that's the question that agencies are always like, is their behavior actually being changes? But it's such a secretive group because it has been historically an adversarial relationship between between management and fishers, and really in a lot of ways, at least from an economic standpoint, the fishers have suffered greatly, not only because of loss of other historic fisheries that were there, but also the increasing number of regulations that they are reluctant to engage, even though many of the fishers that we talk to are conservationally minded. They're just very suspicious of government enforcement, and so we're hoped to be seen as a neutral middle person, sort of providing information into the mix, but they have been they've been tight-lipped on how they're actually using it. It's hard to know how much of an impact, but I would like to think that, I mean, our target was maybe 20 to 30 fishers that are really significantly targeting the bay, and we're getting hits in the 5,000 marks. So I don't know how to read it exactly, but you would like to think that some things are being taken into account. Also, at least the fishers in Delaware Bay they're not targeting sturgeon, they're targeting striped bass. And if you get a big sturgeon in your net, it really ruins your net, right? So they have an incentive to not catch these things. And so but there's also very big consequences for historic examples. Back before the Atlantic sturgeon were endangered, federally listed in 2012, fishermen would report that, yeah, I accidentally caught a sturgeon and I accidentally did this, and this happened on this day. But once they were listed, nobody caught sturgeon anymore, right? So there are definitely conflicting structures that are set up that I actually think inhibit conservation. And so I think there are some structural things and some of those things are historical in nature, but there are some structural things that we need to work on that I do think, in the absence of, militant enforcement out in the oceans, that we could improve on.
So that also raises an interesting role for science, potentially mediating between multiple stakeholder groups. I wonder if either Dr. Carlisle or Dr. Hackrott has also had any of these experiences with stakeholders you've worked with, since a lot of you are sort of working between either government entities and other users of the system or natural resource users. How has your experience been on the ground in terms of being the scientist, collecting information or disseminating it? Have you run into similar conflicts? And what role do you think science has been playing?
Sure. Yeah. No, this is as Dr. Oliver and Dr. Hackerott, I'm sure we'll both say, is like, from a scientist point of view, a researcher's point of view, my own experience has been that you get the different sides, right? You get the kind of the stakeholder in terms of like the anglers or the fishers who view it as a resource versus kind of more conservation side of things. And, most of the pushback I've actually gotten for the kind of work that I do is from the conservation side, where it's the same kind of thing where you're just showing them where to catch this animal or doing whatever. And or, if you put a tag on an animal, you're going to hurt it. Is it impacting the survival of the animal? And those are kind of the animal welfare thing that's been a bigger issue for me. personally is just, but we have to go through very extensive animal care protocols, show that we're kind of doing all of our due diligence, doing everything to maximize survivorship, minimize the impact on the animal. But there's this inherent idea that, we're describing things, processes and dynamics that anglers and fishing fleets and fishers know already are going on out in the ocean. And so like usually if I want to figure out what's going on with a species, I'll just go talk to a fisher, an angler, and they will tell me. And so it's kind of like we're providing, I feel like a lot of times the data we're using and generating are more useful for the enforcement or for management because they don't have that information. They don't know. But it's always going to be there's a trade-off, and everything is a trade-off. The more information you put out there, there's pros and cons with it, and it depends on how people use it and how responsible they are, how ethical they are, what their goals or objectives are. Um, but it's definitely a consistent like just dynamic that goes on in terms of, well, is this helping or not? And in a lot of basic biology, it's like, well, or ecology work, we'll say this helps conservation and management in everything we say, but it's like everything we write. But the reality is how do you actually transform these data and this information into something that actually helps with management? And there is obviously, like as Dr. Oliver mentioned, there's a lot of pushback from a lot of stakeholders because again, any regulation is bad regulation from their perspective, and I sympathize with that to a large extent. But I'm sure, I mean, this is something that I think Dr. Hackerot certainly has probably a lot more experience on the ground dealing with than I do. So I'll stop there.
Well, with my work, it's not as much on the enforcement side, but we do have to navigate that, especially in the Pacific, where depending on the country that you're working in, the reef could be managed by the government or the individual village or even a single landowner. And so I think it's really important to build those relationships with the communities that you're working in, and especially in those other countries, more asking what you can do to help based on their priorities and hopefully helping to guide those priorities a little bit based on our scientific background and that conservation lens. But nothing is going to be very efficient if we're just imposing what we think they should do. It really has to be that ground up approach. And it takes a while to build those relationships so that there is that trust, especially with showing their backyards and giving you access and being open to considering different types of MPA restrictions or fishing method limitations that could help protect physical damage to the reef and things like that. But I think it all starts if you have that relationship built first.
Yeah, that's a great point, Serena and Dr. Hackerot, sorry. And one of the things that we found, and this is back to Dr. Carlisle's point, is that the folks that are out there every day actually fishing, they have an understanding of the environment that I wish we had. There is a level of knowledge, I guess you could call it indigenous knowledge, where they are the ones that have grown up in the area and have been out there since they were kids, and their parents have been out there since they were kids, and their parents have been out there since they were kids. And the amount of knowledge that they have really is astounding. And when you actually get to know them and talk to them, they know that things are wrong. Like they have enough history in their family with their friends to see how things used to be and how things are now, and they're frustrated with it. And so I don't think that we should think of these people who are occupying these coastal, even open ocean spaces as if they're just totally oblivious to anything that's happening and they're just going out and catching their fish and having their day. They are deeply concerned about the environment and their livelihoods. And so it would seem to me that there could be some immediate, but they're also bound because this is how they're economically tied to the system. And so I'm seeing nods, right? And so I do think and this is not my expertise, but I do know that we do pay farmers to not farm. Right. And I do think that we could do something similar in these situations. We could pay people to not fish, or there are different strategies to get them on board, or at least then to get them into a different economic framework to where they are rewarded not only for their conservation activities. And that, I think that's an open world that we should explore as a global community. And so yeah, I would just want to emphasize that the fishermen that are out there, they know a lot. They know so much about where these animals are going. And just like Aaron said, if you really want to know what's going on, go hang out at the docks for a couple days and you'll figure it out. And they may not describe it in exactly the terms that we need, but once you get to know them, the information is there. And I just think they're hesitant. And it doesn't mean that there aren't bad actors, right? So you get into that fishing community, they'll say things like, look, we were doing great until these two fishermen over here went over there and knowingly broke the law, and we tried to warn them, and now we have this new regulation on all of us that we're upset with, right? And so that inside that community, it's hyper diverse about how they approach things and who's going to play by the rules and who isn't, and they all know, they know what's happening. So, but it would seem like it's on our end also to think about putting them into another economic framework so they don't feel like that they're facing economic extinction, every time a new regulation comes out.
And real quick, just to follow up, because I think this is really interesting, but it's like from the fisheries point of view, especially in kind of the angler community, when processes like the creation of marine protected areas start happening, and there's these feedbacks and dialogues between stakeholders and management and whatever. Without fail, the ones that are the most supportive generally are the commercial fishers, the commercial anglers that are very invested. They view it in the long term. They're interested in maintaining, they want to help. They want to make sure that these resources are there into the future. The ones without fail that are the most vociferous against any kind of protected areas or regulation are the recreational anglers, which are a huge stakeholder in the US at least. And they just view anything that limits their ability to get out there as being very bad. And they kind of view, hey, this is my right. This is my ocean. I'm not the one that's causing the problems. And it's the commercial anglers that are the commercial fishers that are ones that are really driving everything, which increasingly is not the case. We have good data that show that there's so many recreational fishers out there removing so many animals or interacting with so many animals compared to the scale of commercial fisheries that they have a huge impact, and they just don't think about it. And so I've always been surprised that it's the recreational fishing community that is the strongest, the biggest barrier oftentimes to effectively implementing management practices.
Well, I thank this panel so much. We are at the end of our session. Thank you, participants, for coming. Please follow up with our speakers. Their information is posted on the Virtual Ocean Pavilion website if you have any further questions. Thank you so much.
Thank you, everyone, and thank you to our panelists and to the University of Delaware for organizing this amazing event. A recording of this session will be posted to the Virtual Ocean Pavilion platform, as well as the Global Ocean Forum YouTube channel. And we encourage you all to check out our many features available from November 6th to the 22nd. Extending our best wishes on behalf of the co-organizers of the pavilion this year, the Global Ocean Forum and Plymouth Marine Laboratory. Thank you very much.
Thank you, everyone.